Okay, good afternoon, everyone. For me, it's a pleasure to be here and to present this collaboratory work named Epithelial Mesenchymal Transition Signature Influence Patient Outcome in Colorectal Cancer. Is a group effort from this diversity institution in Latin America, and I'm in representation of all the group. So colorectal cancer is ranks like the third most frequency diagnosed malignancy, like we see right here, and it's the second highest cancer-related death. And like we know, in similar in other solid tumors, metastatic neoplasies are associated with poor survival. In this case, since epithelial mesenchymal transition is a key process of the tumor metastasis, we use single-cell data to evaluate relevant signals for transitional phases in epithelial tumor cells. So the first thing that we do was to use a data set that includes this anatomical part of the cells, and then we try to distinguish between normal adjacent cells and tumor cells. And to distinguish these cells, we use infer CMV to know to infer the copy number variation, and we just differentiate between normal adjacent cells right there and the tumor cells right here. In the second step, we exclude the normal adjacent cells and cluster epithelial and non-classified cells. Like we have this amount of cancer cells, and also we use only the sigmoid colon cells, and we cluster into 15 subpopulation, and all the cells we cluster in 22 subpopulations. Then we want to know what is the relationship between this cluster and the EMT markers. So we do a literature-based gene list to determine that we have 4 clusters related with the EMT process, and in the sigmoid colon cancer cells we have 2 clusters related with this biomarker. We explore like different types of process, like macrosatellite instability, the canonical subtype, the metabolic subtype, and the mesenchymal subtype that was the CMS4. So in this case, we have 4 clusters right there and 2 clusters in the sigmoid cluster. So when we found these mesenchymal markers in these 2 clusters and the 4 cluster, we try to know what is the expression of these mesenchymal markers in this cluster. So we have we found 20 differential expression genes in these EMT putative cells. That was the cluster 8, 10, 17, and 21. And then in the sigmoid colon EMT putative cells, we found the cluster 6 and 11 that have high expression of these mesenchymal markers. So then we want to know if there is a relationship between the evolution of these epithelial cells and these clusters that express mesenchymal markers to evidence the EMT transition. In this case, we perform a pseudotemporal trajectory analysis in all the cancer cells and in the sigmoid colon cancer cells. And in this case, we infer only a differentiation between the evolution of the epithelial cell to tumor cells in the cluster 6 only. In here, in the 4 cluster, we don't evidence so much this transition to epithelial to mesenchymal cells. And in this, all of the 20 differential special genes, we only found one gene related with EMT markers. That was the BIN gene. That it was more expressed in the mesenchymal-like cells compared with the other cells. Then we want to know what is the pathways related with the EMT markers. So we perform a gene ontology enrichment analysis of all the differential expression genes, also in all the cells and the sigmoid colon cells. And in a broad spectrum, we found that in above 30% of all the EMT putative cells have an enrichment for activation of relevant pathway like cell differentiation, that's right here, developmental process, and also cell adhesion. So this pathways is very related with the EMT transition. Finally, we want to know if there is a relationship between the treatment in different data sets of colorectal cancer and markers of EMT transition. And we know that of the differential expression genes between the responders and no responder of two types of treatment, this one was FOLFIRI of these data sets, and the other one was the treatment FOLFOX in this data set. And in the first data set, we found only three genes related with the EMT process. That was the 2484, that's PARCL1, and the CRAC CR2B. And all these three genes are related with differential expression between responder and no responder. In the case of this PARCL1, the relationship is inverse because it's a gene related with modulation of the cell migration and invasion. So is expected to find in no responder a lower expression of this gene between the responder. And in the other data sets, we found that this gene, the PP1R40D, was differential expressed between no responder and responder, and this gene is a EMT marker. So this is a first approach for this study. We want to know like more to use more data set related with the colorectal cancer. And I want to thank you all the collaborative work from Daniela, Silvana, Anne, Patricia, Alexis, Cristobal, Dave, from this all collaborative Institute of Latin America. And I want to say thank you to the ESCB for the invitation.
Thank you for your presentation. Okay. So you know probably what I'm going to ask about the functional analysis. So you show the analysis for the gene ontology, right? Have you done the analysis with other more specific databases like Kang or Reactor?
We want to do more analysis because this is a first approach for the analysis of colorectal cancer. So thank you to the tools that you teach us in the pre-course of Congress. We want to do more analysis and more integration of different data sets because we try to filter the data with the infer CMB first, but it will be like a more depurate the conclusion that we have if we maybe incorporate other approach like proteins, like microRNAs and all this stuff.
Mm-hmm. Yeah, I have a question about your last slide.
Yes, this one.
Yeah. So, how did you pick these four genes? Were these the job of the differential expression, or what?
We compared the responder and non-responder, and we obtained a total of 23 differential expression genes. But we want to notice the genes related with the EMT process. So of the 23 genes in these data sets, we only obtained three genes, literature-based gene list, related with the EMT. And in this case, in the other subset, we only found one gene of 5 differential expression genes.
But in terms of significance, were did you have like genes with a stronger p-value corrected, or
Yeah, it's a p-value corrected, right?
yeah? But my worry is that the significance is here just one star.
Yeah, exactly. It's like a first attempt because we need to incorporate more studies, but because it's a started project, so we know that it's just one data set. We need to incorporate more data sets to validate these findings, but we hope to do in the next month. Yep.
Thank you. Nice talk about colon cancer. And have you correlated in the first slide when you spoke about colon cancer? You say that metastatic colon cancer has a very low survival.
Uh-huh.
So my question is, these 4 genes are correlated with survival in colon cancer because this could be a link that you are not having now to the clinical event that is survival. So if these genes mark survival due to expression, we have done some studies on the large series of colon cancer expression profiling of the genes plus a correlation with survival.
Yeah, it's a difficult issue because none of the study have all the metadata that we want to know. Because for one side, there is some study that have metadata of survival days, but for example, in this study, it's only the metadata related with responder and non-responders. So
Yes,
it's
but you can take the time, you can take these candidates that you have there,
exactly.
go into a large
So-
data set
Exactly.
with survival and expression and see if the high expression of these genes or low expression correlates with poor survival or better survival, and this is not difficult to do.
Yeah, exactly. It's like the follow step in the analysis, because it's that we have these 4 genes and we try to validate it with another data sets. But the principle of this lecture is that we are from different country, we are from different institute, and it's the first effort to do analysis between all these because all the person here is from one single cell course in Rio. So for me, it's a pleasure to to demonstrate that we can do collaborative groups internationally and not be in the same institute, and all from Latin America. So I know that we need more to do. It's difficult because there is a different objectives in each group, but we also make it. Mm.
Thank you.
Okay. Last question. Yeah, my question is regarding the data set that you are using. Do you know if there are how many individuals you have in that data set? And also if there is information about the degree of metastasis that you have for those individuals? Because well, maybe it's a question from that also kind of idea. You could make some correlations about the level of expression of some genes regarding the level of metastasis.
Uh-huh. The number of patients like 50 in the data set, but the problem is that the metastatic is just in the metadata. It's just like yes or no. We don't have like a value of metastatic, so we need to explore more data set that maybe we have the value of the or the score of metastatic to maybe to see in the time in the transition how is the expression of these different genes depend of the stage of the metastasis. And also another idea is how to correlate the site of metastasis with the expression genes. Yep. But thank you for your
Thank you.